Okay, so in this video I want to explain how our eyes could detect light signal using these G protein coupled receptors. So inside of the rod cells, which are inside our retinas, are these disc membranes. When light activates one of these receptors, it becomes activated, shown here by R star. And then this allows binding of a G protein. This binding causes the GDP from the bound G protein to become swapped out with the GTP. And once this alpha T subunit is activated by GTP, it then is able to remove this inhibitory gamma and beta subunits from this PDE protein. And so you see now when both of these inhibitory units are removed, the activated alpha beta PDE unit is able to catalyze the conversion of cyclic GMP to GMP. And because there's these calcium channels in the rod cell, which are basically opened by high amounts of cyclic GMP, once this PDE is catalyzing the cGMP to GMP, since there's now less cGMP in the cell, the cGMPs that are bound to the channel will dissociate, and you will have now a closed ion channel. So it will be a closed cGMP gated ion channel, and because of this there will be less neurotransmitter released into the adjacent neuron cells, which carry signals to the visual cortex of the brain. And so basically this shows that light activation of this G protein coupled receptor will allow G alpha T subunit of the G protein to remove these inhibitory gamma subunits from PDE, and then PDE will catalyze a conversion of cGMP to GMP, which will lead to the closure of these ion channels and results in less neurotransmitter release to the brain. And then to go back to the initial state, these GTPs on the alpha subunit have to be hydrolyzed, and that's accomplished by this RGS9 G beta 5 complex, which will catalyze the GTP. And so then that would cause this alpha subunit to dissociate from the gamma, and so now the PDE will go back to its inactive form. Usually this process from light activation back to conversion back to the initial stage could take about 50 milliseconds. And so that explains why sometimes we could see things very quickly. Also, there's this right here, this diagram showing that depending on the amount of time that these receptors spend in light, they could be inhibited by these rhodopsin kinases. And so you see here, when at the initial time the receptor is activated by light, and here it has full activation represented by these arrows. At some point further in time, rhodopsin kinase will phosphorylate it, so it attaches a phosphate group to the C-terminus. That reduces the activity. And then even further in time, a second phosphate will be attached by the kinase, and then this allows an arrestin protein to bind to the receptor and prevent it from activating any further visual signals.